Hello, and thank you for joining us today for the webcast, Transforming Cancer Treatment with Early Detection Diagnostics, Opportunities and Obstacles. My name is Afamia Murray, and I'm with the marketing team here at Pharma Intelligence. Before we jump in, let's go over some housekeeping items. First, if at any time you are having technical difficulties, click on the Test Your Connection button, and this will take you to a connections checker and FAQ page. If you are still experiencing any issues, there is a chat now function in order to go through to the support team who can assist. Want to enlarge the presentation? Just click on the maximize icon on the lower left of the presentation window. There will be a Q&A at the end of the presentation. So if you have questions, we want to hear from you. To ask something to our presenters, type your question into the question window toward the bottom of your screen and then hit submit. We'll be answering as many questions as possible during the Q&A session. And if there's extras, we'll answer them for you via email following the webcast. Because we know you'll want to hear this webcast again, today's session is being recorded and will be available to view on the on-demand following the end of the live event. Now, I would like to introduce our moderator, Ben Comer, Executive Editor at InVivo. Ben will be filling in today for Phil Greenfield, Editor-in-Chief at MedTech Insights. And with that, I'll hand things over. Ben, the floor is yours.
Excellent. Thank you so much, Afamia, and thank you everyone for joining. I want to make just a one change to the Q&A. We may not have time to have a formal audience Q&A at the end of the session, so I would encourage anyone with questions to submit those during the webcast, and we will try to answer as many of those during our discussion as we can. And I also say that you're free to email me afterward if your question is not answered, and we will respond. Now, I want to introduce our very accomplished panel of speakers. I think each one comes at this issue from a unique perspective. Our first panelist is Dr. Azra Raza. He's the Chan Soon Shiong Professor of Medicine and Director of the MDS Center at Columbia University in New York. Dr. Raza is an international authority on pre-leukemia and acute leukemia, and supervises a state-of-the-art basic research as well as seeing patients. Dr. Raza has been collecting blood and marrow samples on her patients since 1984, and her tissue bank now includes over 60,000 samples. She's also the author of The First Cell and the Human Cost of Pursuing Cancer's Last, a book that was published by Basic Books last October. Our next panelist is Harris Kaplan, a managing partner of Red Team Associates and CEO of Healogics, a life sciences marketing research company. Harris has worked on the introduction of more than 100 new products, including drug, biotech, device, and diagnostics. Harris is a commercial strategist and an advisor to a number of VC firms. He's also authored articles for several industry publications and sits on the InVivo editorial advisory board. Our next panelist is Helmy Eltoukhy. He's a co-founder and the CEO of Guardant Health, a leading precision oncology company with a mission to conquer cancer with data. Under Helmy's leadership in 2014, Guardant Health launched the first comprehensive liquid biopsy. And in August of 2020, the company's Guardant 360 companion diagnostic became the industry's first FDA-approved liquid biopsy for comprehensive genetic profiling. Helmy holds a PhD in electrical engineering from Stanford University and was a postdoctoral fellow at the Stanford Genome Technology Center. Last but not least, we have Sam Esgarian. He is the Chief Medical Officer at Thrive Earlier Detection and will lead medical strategy, including the integration of CancerSeek into routine medical care. Prior to Thrive, Dr. Esgarian was Vice President of CVS Health's Transformation Health Product Organization, where he was responsible for the modernization of clinical products and implementation of new solutions to improve consumer convenience. Dr. Esgarian holds a BA in molecular and cell biology from UC Berkeley, an MA in medical sciences from Loyola University Chicago, an MD from Tulane University, and an MBA from Cornell University. Thank you all panelists for joining. Before we jump into our discussion, I want to just share a few highlights pulled together from my colleagues at MedTech Insight and MedDevice Tracker. The molecular diagnostics market was initially limited to hematological cancers, but now has expanded to include oncogene testing for products targeting HER2, KRAS, BRAF, EGFR, and BRCA. New molecular cancer markers are being discovered regularly. There are now over 165 oncology biomarkers that were approved by 2019, and now 90% of pivotal trials involve molecular targets. 43 companion diagnostic products for non-small cell lung cancer are in development or have reached the market, a market worth around $1.35 billion in 2009, according to Informa's MedDevice Tracker. Lab-developed tests, including companion diagnostics and precision diagnostics, are driving growth in the molecular oncology market. More than 335,000 Exact Sciences Cologuard colon cancer screening tests were reimbursed for Medicare patients in 2018 with payments of more than $170 million. Lab-developed tests often migrate to commercial test platforms and in vitro diagnostic test kits, and that trend is expected to provide ongoing stimulus for the in vitro diagnostic molecular oncology market going forward. Liquid biopsy is a hot area for investment and deal making. There have been a number of IPOs, funding rounds, including Thrive Earlier Detection with a Series C raising $257 million, and lots of M&A activity that I won't go through here. But I will make a final point that cancer care is the fastest growing cost in U.S. healthcare, estimated to reach $158 billion in 2020, which represents a 27% increase from 2010. And with that, we will get into our panel discussion. And I want to start with Dr. Raza to help set this discussion up with the basic question, which is, why is early detection needed?
Well, thank you very much, Ben, and to the In Vivo organizers for giving us this opportunity for me to join such an accomplished panel that you have put together. Congratulations. So when my credentials really come from the fact that you mentioned, which is that I am seeing 30 to 40 cancer patients every week, and I've been doing that for the last 30 plus years. I supervise a basic research lab, which is, and we have a lot of publications, of course, but I think my most important credential is that I'm a cancer widow. So I've had the chance to see cancer from all kinds of perspectives, including both sides of the bed, as a caregiver and as someone whose loved one is dying of cancer. I don't wish this perspective on anyone else, but the sad thing is that I started in this country my career in oncology by treating acute myeloid leukemia with two drugs we popularly refer to as 7 and 3, 7 days of one, 3 days of another. Today in 2020, 43 years later, I'm still using 7 and 3 to treat acute myeloid leukemia. Yes, of course, we can cure 67% of all newly diagnosed cancers, but it's not because necessarily of great treatment. The 1% decline in mortality in the last 30 years has really come from anti-smoking campaign and earlier detection. And the 67% we cure are because of these two reasons mainly. Now the 30% who are still dying of cancer today are diagnosed with invasive advanced disease. So if we think in a universal way, there will be 10 million people dying this year from cancer worldwide. And a good 80 to 90% will die of advanced disease. So then it's no surprise that early detection can be curative for a lot of patients. And this is nothing new. It has been known since 1907 when Dr. Childe said that it's not cancer that kills, but it's the delay in treatment that kills. Okay, so why are we not making more attempts for early detection? We have been trying to do screening of individuals, but what we've been doing is testing one person, for example, for the most common cancers, colon and lung and breast and prostate, etc. But there are a lot of people dying of uncommon cancers also. A person doesn't get to choose their cancer. So how do we make this more universal? Clearly, if early detection is needed, the screening measures we have put into place so far have only been, let me give you some numbers to put this into perspective. What I am saying is that with the current measures, we are spending something like, in America, $27 billion for screening measures. And we detect 9 million positive cases. But out of those, do you know that the real cancers are only 200,000? 8.8 million are false positives. So this is not a great ringing endorsement for early detection of cancer, but that's using the ancient archaic techniques. What we need is more sophisticated technology and improving early measures so that we can screen healthy individuals, as cancer is, silent killer. We use the sophisticated molecular genetic markers in screening healthy individuals to find illness before it has become a bona fide clinical disease and prevent it. That's what we need to do. And the second thing we need to do is bring multi-cancer early detection test, the MCED, which means that from one sample that we get from a patient, instead of looking at one type of cancer, we can look for multiple types of cancers. So this is my plea to everybody. Early detection is very important. It's the only thing that has been shown to save life, except for very small steps in terms of improvement of therapy. We are still using the Paleolithic measures of slash, poison, and burn. And that's gotta stop. And the only way to stop it is early detection and prevention. So I will stop here.
Thank you, Dr. Raza. And I want to turn to Sam next on this question of why early detection is needed. Dr. Raza mentioned a single sample testing for multiple cancers. What would you add to this question, Sam?
I could be very brief, but we have time. I can say all of the above because Dr. Raza captured it so perfectly for what we envision for the future of cancer prevention and cancer treatment. At Thrive Earlier Detection, all of our science is founded by what Bert Vogelstein and the teams at Johns Hopkins have also agreed upon, which is we can go after treatment, but the real value for cancer is going to be in prevention and detection. And now with the advanced technology that we have, before we wouldn't have been able to do this, right? But today, can I take a sample of blood from a patient and surveil that sample for the signs of tumor DNA that's in that bloodstream? And now I'm actually detecting cancer. And can I do it in such a way, because it's a screening solution where Dr. Raza was spot on, the specificity is as high as it can be. I'm ruling out false positives. I'm calibrating the volume so that I'm finding cancer, and I'm eliminating the noise of inflammation, of other things that could be going on at a cellular level, but is not cancer. And what it does, and what we love about this, thank you for having us in vivo, it really does flip the script. The script right now is exactly as Dr. Raza described. It's sick care. It's secondary and tertiary care beyond the reach of a primary care provider. And so how do we use this technology? How do we take something as simple as a laboratory test and put that in the hands of a primary care provider? It still is in the hands of a physician and a clinician who can really work with the patient and the population that he or she knows so well. But now they have the tools to say, well, I was diagnosing diabetes, I'm diagnosing allergies, I'm diagnosing all these other diseases and illnesses. Now I have the tools to do that same thing and apply that towards cancer. And I'm not going to treat it. Dr. Raz is spot on, but I'm going to coordinate the care from this point of view and then allow the specialists to really see this at an earlier stage where the treatment can be curative. And now I'm looking at it, as she so well noted in her book, at the first cell versus the last cell. And in Thrive, we look at it exactly the same way. How do I downstage this so that you're looking at it from stage 1 and stage 2 versus what you see today, which is stage 3 and stage 4? And can we do this in the way that a patient would expect us to do this? This is the last part that's super critical. A patient would expect us to look for cancer. They don't look at it the way that physicians and clinicians do. They're not saying, Sam, I think I have lung cancer when they come into the clinic. They're saying, Sam, I think I have cancer. Can you go and surveil and find out if I do? And that multi-cancer approach gives us the ability to treat this in that broad spectrum and in that broad spectrum, narrow it down, and then let it be treated at that individual organ and distinct level. And that's really kind of the premise for what we need. And one last part of this is the treatment part that Dr. Ross brought up. That's where we wanted to focus our efforts. That's where the folks at Johns Hopkins could have gone after treatments, but we've almost hit a plateau in that regard. And the treatments haven't changed much. And most importantly, the outcomes haven't changed much. So what do human beings do when they've reached that type of plateau? They look for a breakthrough. How do I look at it in a different way? And instead of constantly focusing my attention on researching further treatments, the breakthrough that we believe that we're working towards is looking at the screening and the prevention upstream to then help filter out the outcomes that we expect, a good outcome on the treatment side.
Excellent. Thank you, Sam. I want to move on to my next question, and I'll start with Helmy on this one, which is, what kinds of technologies are emerging for early cancer detection? And secondly, how do these technologies compare with advanced screening detection tools like blood-based genomic profiling for patients with cancer in advanced stages?
Yeah, that's a great question. Let me start with the second half of it, which is what we do at Garden today, which is help stage 3, stage 4 cancer patients essentially get the best biomarker-driven therapies. And a lot of what that technology is built on is looking at what's called cell-free DNA, or DNA that's being shed from these tumors into the bloodstream. The problem is the tumors make up a very small amount of the overall cellular mass of an individual. And so this cell-free DNA that's there in the blood is found at very, very low concentrations. And so what our test does, Garden 360, is that it's able to detect these, very low concentrations of cell-free DNA and sort of reconstruct the genomics of the tumor in those patients. So then we can match the mistakes in that genome, the mutations that are there, with the best possible therapy. The technology is at a point now where we can detect single mutated molecules like an EGFR mutation or insertion and deletion in a tube of blood. And so what we found, though, is that just looking at those alterations in the sequence, the ACGTs, whether it's substitutions, deletions, amplifications, fusions, things like microsatellites instability, looking at those at extremely low concentrations is unfortunately not enough to get to very high sensitivity for stage 1 disease. If you think about the tumor itself has mutations that and changes that define it. But there are other changes that happen, not just in the tumor, but in the microenvironment as well, that could lead to incipient disease. And those are kind of multiple levels. So you have the mutations that are there, which we look at it with single molecule sensitivity. But then there are chemical changes in the DNA itself. There's methylation, there's hydroxymethylation, there's other changes as well. And those changes often pervade the tumor itself. They're often found in the microenvironment, and we potentially have much higher detectability of those changes that are there, especially in early cancer detection. So we're seeing companies like ourselves, augment what we see on the genomic side with this epigenomic dimension, looking at beyond just the sequence of letters in the genome. Furthermore, there are other changes that happen. There are structural changes. The DNA itself is wound around these histones or nucleosomes, sort of like, basically yarn around a spool. And the spacing of those spools or those nucleosomes changes depending on expression of the genes and from cell types to cell types, from cancer cells and normal cells. And so we developed a technology for our early detection program that looks at those three dimensions simultaneously with very high sensitivity. There are other biomarkers that other companies are exploring as well, such as looking at further into the epigenomics domain, into RNA level, so looking at essentially how the DNA is transcribed. And the beauty of looking at things kind of in that later cascade is, RNA, you get a sort of biological amplification. If the gene is expressed, you get multiple copies of RNA for every single copy of DNA. And then, other companies are looking at proteins as well in the blood, and you get further amplification there. So, the blood is, what we found is an extremely rich landscape for, these changes that happen. There's heavy communication between all of these, different compartments that are there. And it's really just been fascinating to see how much headroom there is in terms of really augmenting the science that exists today in terms of how early and how specifically we can detect cancers. As you start looking at these epigenomic changes, you can also more easily classify the type of cancer that an individual has, and really think about things like tissue of origin and so on. So it's been a fascinating journey, I think, in the field in general, and really been gratifying to see how much more investment there is in early detection than there was when we started the company 8 years ago. I think one last thing I'll say is that I think when we started, there was probably $90 million total of NIH funding to early detection out of tens of billions of dollars. And now you see these funding rounds, with Thrive and other companies and ourselves, and it's been really gratifying to see that investors really do appreciate the impact that early detection can have on the space.
Great, thank you, Harris. What would you add, what kinds of technologies are emerging that are interesting or exciting to you? What would you add to this question?
Well, I'm not so sure, to be honest, Ben. I'm not as sure that I'm the right person to address the technology issues, but I do think that there are some challenges. I think everybody on this panel needs to be grateful to companies that have kind of led the way, like Diagene, with the early detection for human papillomavirus, and Exact Sciences, which was mentioned briefly. But, the reality, and I think, Ben, you mentioned, my background is commercial, right? Even now you can find article after article pushing back against Cologuard, as an alternative to colonoscopy. And so I think the issue is that we've got technology meeting reimbursement headwinds, right? Of how do these things really come together in terms of the value that they can bring, and who are the appropriate candidates. And Diagene and Exact Sciences are both for clients. And with Exact Sciences, I can tell you that people look at them and say, well, the glass is only 90% full relative to colonoscopy. And I look at it and say, look at all the people that won't get a colonoscopy but are willing to do a Cologuard, right? And I think that, shifting from the technology a little bit to how do we make early screening a pragmatic and financial reality, is a question I'd like to throw out, to Dr. Azra and Helmi and Sam. because I think that's a challenge. Everybody wants earlier detection, but how do we actually get there?
Yeah, absolutely. And I'll just follow that by asking how early detection technologies would change the treatment pathway, flip the script, so to speak, for cancer patients, and potentially improve outcomes. And I want to get back to the issue of some of the obstacles, Harris, on reimbursement, as well as drug development issues. So we'll come back to that. But what about the treatment pathway for patients? How does that change, and how does it potentially improve outcomes? Dr. Raza, do you want to start there?
I would love to. So listen, Ben, I have spent so many years thinking about exactly this issue, that why do we have this spectacular failure in cancer treatment? And I call it the CRUSH syndrome. CRUSH is an acronym I use, that we are trying to address a very complex disease, whether you look at genomics, proteomics, transcriptomics, metabolomics, and in different compartments of the body giving different signatures. Basically, the answer is the farther you go into the sea, the deeper it gets. We have learned a tremendous amount about the biology of cancer in the last 50 years. But have we been able to translate that into improved therapies by the bedside? Very little. This is my problem. So the CRUSH is complexity of cancer being addressed by reductionist approaches, and then making an ultra hype about minor advances in mice. And of course, the other problem being simplistic clinical trials. By the time we diagnose cancer, the cell has so many mutations, and we are coming after one targeted treatment. All of these treatments that are failing today, 95%, might be more successful if we could diagnose early. And the H in CRUSH, the last one, is the high fiscal cost. This country is spending $3.5 trillion on healthcare every year. It is on the verge of a financial collapse already. So we can't even afford it. It's unconscionable that 42% people who are diagnosed with cancer today in America lose every penny of their life saving by 2+ years. It is simply obscene. We shouldn't be doing it. So the answer to your question about how can this early detection improve treatment is, I think we need to get away from the whole treatment paradigm and go towards prevention with early detection. This is the whole point, that the earlier you detect a wellness to disease transition, you should be able to detect it years in advance. For something as revealing as cancer is in its early footprints. And then I think that we shouldn't try to fit a horseshoe that made a horse run faster on a car to make the car go faster. We are not going to use ancient technology to deal with the first cell. Many people ask me this, Ben. Well, Dr. Raza, you are saying we should be finding the first cell. What are you going to do if you find one cell in a cancer cell in a patient? Give them a bone marrow transplant? No, of course not. I'm not going to give them a bone marrow transplant. But earlier on, the disease is going to be less complicated, perhaps. We know the current strategy is not working. So we need to go to newer strategies. And they will co-evolve with the biomarkers we detect and the more targeted therapies we can bring. So I'm very optimistic, in fact, that the earlier we go, the better treatments will become available, but not even treatment, better measures for preventing the disease. And I'll finish by saying that what I'm trying to do is, you mentioned that I have 60,000 samples. Yes, on thousands of patients who have marched from pre-leukemia to acute leukemia, or to die of bone marrow failure, and I have longitudinal samples on them. What I'm doing is multi-omics, proteomics, transcriptomics, metabolomics, and the methylation platform. I'm working with GRAIL, for example, to do the entire tissue repository with these multi-omics, find what are the earliest footprints, and try to do multi-omics on single cells. So when we start finding the first cell, we will be able to look at the heterogeneity of individual cells within that clonal population. And for all of this, we need the competition of the space age, we need the cooperation of the Human Genome Project, but above everything else, like infectious disease folks did it, by inventing vaccines to prevent the disease. That has brought the most dramatic revolution in medicine, is prevention by, whether you look at cardiac disease or infectious disease. Cancer has to be the next, and it will only happen when we can find the earliest footprints. And that's your answer for the treatment.
Yeah, and I'm glad you mentioned the issue of cost, particularly at end of life for cancer care. It's a very difficult issue for families. As you know, we've heard this phrase financial toxicity emerge in talking about specific cancer drugs. But Sam, I want to just turn back to you and ask how Thrive is thinking about a new patient journey, associated with the kinds of products you're developing.
I think the key is, and I want to change it just a little bit, we're not thinking about it, because thinking about it is so theoretical and hypothetical. What we wanted to do is go out there and measure it, because I can sit here and say, here's what I assume may happen. And, here are a series of assumptions, and here's an equation to it. The reality is it's so disparate, right? Is it in a rural setting? Is it in a metropolitan setting? Is it with telemedicine? Is it in an on-site center? Is it an integrated delivery network? And so our goal is to go and test and measure this out, right? One of the benefits of what I'm already saying is we've started to. The DETECT-A results that we published this year was a seminal study in that for the first time we looked at a prospective interventional study that went out there and did this with 10,000 women aged 65 to 75. And we had two key outcomes we were hoping to measure. Can we detect cancer early enough so that the treatment is curative? And can we do it in a safe way? Can we do it in a way that is complementary to the care that they are receiving and is not a replacement? Because the key here is I'm going to disrupt the provider's workflow. I'm going to introduce something new to a primary care provider who's never done this. And so what happens? Can they continue to maintain that close relationship with their patients? Look at all the comorbidities that come in. A 65 to 75-year-old population, surprise you, every single one of the people enrolled had a comorbidity. So are we maintaining their state of health and improving it exactly as Dr. Raza described? Am I getting to health and wellness and that well care versus the sick care of today? And the study was successful. So now our attention is to continue to test and measure this. How well do I perform in a multi-cancer scenario? Working very closely with the FDA and saying, how do I design a pivotal registrational trial where I can go and do this and do it across the multi-cancer aspect? And the best part of all of this from a treatment pathway and a cost pathway is I'm generating outcomes and I'm measuring those. I'm generating real-world evidence and I'm using that to validate it and I'm feeding that back in. So what do we do with a machine learning classifier around the cancer-seq test? Is we continue to calibrate its volume and continue to increase all of those metrics that are critical to its success. And that's, you could use any other industry. That's product and how a product enters. Now we're being extra cautious for a healthcare product. We're a very new, unique, innovative healthcare product. So our goal is to do this in collaboration with our provider partners and in collaboration with patients, with individuals, with advocacy groups, and really kind of deploy this at scale in a manner where we've already figured out the math behind it, and we're happy to build it and deploy it. That's the best way. That's at least the way that we would like to pursue figuring out how it impacts the treatment, how it impacts the reimbursement plans and benefit designs of health insurance and the provider.
Thank you. I want to move on to address some of the challenges associated with early detection. But there was a question that came in from the audience that I want to direct just quickly to Helmy and Sam first, which is the question is, given that these new biomarkers are being discovered, are liquid biopsy early detection assays like Guardant's and like Thrive's also reassessing their own technologies in light of these new data? Are new technology and assays amenable to these new discoveries. Maybe Helmi, I'll start with you, and then Sam, I'll give you a chance on this one too.
Okay, yeah, I just wanted to kind of maybe address some of the previous comments. How can we do better than we are? Just a couple of weeks ago, my brother-in-law, he's 36, got diagnosed with stage 4 cholangiocarcinoma, and it's just a terrible situation. So it's I think to Dr. Raza's point, the bar is pretty low. We can do a lot better in terms of diagnosing much earlier, and fortunately, we've been able to, he's TMB high. We've been able to, get him onto, Keytruda. But to Dr. Raza's point, we're essentially trying to stop a fire that's already burned down a house and trying to salvage, the situation versus finding essentially incipient disease. I actually think you have to go beyond just finding cancer as early as possible. I think where we found that early detection works most significantly is when we find precancer, when we find essentially the tissue that is really has predisposition to the start and birth of different cancers. And I believe that with the technologies we're working on and with the science happening in the space, that will happen, and we can go from essentially post-cancer treatment to pre-cancer treatment, prophylactic treatment, where we reduce inflammation and reduce the birth of cancer in the first place. That's the ultimate goal that we have as a company. In terms of, the technologies that we're seeing, it's really fascinating. We call, our tagline is "Conquer Cancer with Data," and it's because, to Dr. Raza's point, is we just need a lot more data. There's been, I think, some arrogance in the space, in general in scientific field about, our thought that we understand the biological complexity. Complexity is extremely high. We have extreme intellectual humility as a company. And so we have, I think, following in Dr. Raza's footsteps, we have almost 200,000 samples in our biobank now. And we go in and do the same thing, deep whole genome sequencing and so on. And I think that's the only way that we can get there. And so, it's not just the technologies we're working on. There are thousands, looking at the immune system, looking at the amplification there as well, as another, area that, all of these things have to come in. I think eventually it's not just blood-based technology. It's going to be radiographic, technology. It's really a confluence of all of these different modalities that are going to work towards, finding pre-cancer or early cancer, really as early as possible.
Yeah, and biometric sensors potentially as well. I've seen some products in development along those lines. Sam, I want to give you just a chance to respond to this issue of how you incorporate new discoveries like biomarkers into your ongoing development plans at Thrive.
Yeah, I think we're in this world where the technology continues to speed up and we continue to adopt it, but learn how to use it in a way that's most effective, right? There's many buzzwords in healthcare today, right? The three most common ones are transformation, machine learning, and CBD oil, right? These are the catch-alls. I'm going to solve the problem in healthcare with one of these three things. But what we're really seeing is, can I do this faster and effectively, and can I test it out and then deploy it and make sure it works as I tested it out in the laboratory? So one of the things that we have is that safe sequence technology that we've licensed from Johns Hopkins. It's already become the safer sequence technology from a next generation sequencing standpoint. So our turnaround times are fast, right? I can start to analyze this and get these results into the hands of a primary care provider sooner. And then once I have those results in their hands, so I can start to validate how well did it work. And as we look at it from a biobank standpoint too, I wasn't doing short shrift, right? There is a software machine learning component. And that's going to start to help us because it really is the genetic code now. But it's going to be what everyone says it's going to be. It's going to be a zip code. It's going to be all the socioeconomic factors. How do I not look at it at the cellular level, agnostic to the individual level, right? Where did the cell come from? Where is he been living? What has he been doing? What's his lifestyle been like? And how do I continue to analyze that, not just in our lifetime, but in a very quick 2-week turnaround time, to then get a result back to a primary care provider and allow him or her to make the decision for what to do with this result, right? How do I then make it confirmatory? And once I have that confirmation, how do I coordinate the care into the right hands of the right specialist in an effective manner? And you've seen this, right? Now I'm starting to talk about getting to treatment versus getting to a diagnosis. And that, to answer your question, is the fastest path of what we want to use the technology towards. How do I solve the problem that we spend so much time when time is so valuable to the individual? And then use that time more importantly towards the treatment and the outcome, and then putting them back towards a higher quality of life that they want to enjoy. That's all.
Great, and I've seen some questions come in about public awareness and education efforts, but before we go into ideas about that, I want to ask about some of the obstacles. And first, I'll ask, are there any risks associated with detecting a cancer too early? For example, what if a cancer screening is positive but requires follow-up scans or imaging, or can't be easily located? Dr. Raza, do you want to take a stab at that one?
Yes, because this is an issue we deal with all the time, Ben, and it's a very important issue that you have raised. And the biggest concern always is the anxiety it provokes in people to have some kind of suggestion that you may have cancer. And I want to point out that it's not going to be an issue only with early detection. I gave you the numbers right now that of the 9 million positive tests we find from screening measures today, 8.8 million are false positive. So imagine, especially a woman who is in her 30s or 40s is suddenly told that there is a mass on your mammogram, and now she has to live through the harrowing experience of waiting for the biopsy or FNA or whatever has to be done to supplement the test. So yes, early detection will also create anxieties, but the idea is that the earlier we find the disease, the more the chance is that we can manipulate for the patient's advantage, even with simple things like changing lifestyle measures or introducing new kinds of diet or programs like that could also be fine for very early footprints. I'm not saying actual cancer, but I'm saying we need to go so far in advance. And at that time, the idea is that things will have to happen simultaneously. That is, the earlier we are going, the better the treatment options should become, and the anxiety should be decreased. However, if today I go and get my blood tested for circulating tumor cells, and they come and tell me, Dr. Raza, we are finding adenocarcinoma cells hanging around in your blood. Next thing I'll do for myself is I'll run to get a PET scan and see which gland in my body is producing cancer. Let's say the PET scan comes back negative. Now what do I do? How many times do I repeat this circulating tumor cell test on myself? And what do I do with it? If PET scan is negative, should I reschedule another one in 6 months? It's going to expose me to a lot of radiation. And all these months, I'm going to be very anxious. So I think Helmi is absolutely right in mentioning, and so is Sam, that it's got to be a combination of biomarkers, devices, scanning, imaging things. We are at Columbia, we have been working on a chip with the biomedical engineers. Sam Sia is the professor. This chip is already FDA approved in America you just put a drop of blood here, and through microfluidics, it travels and it can give you the PSA level sitting at home. You just have to put this chip in a device that comes with it. It's FDA approved. My point is, if we can have a biomarker for ovarian cancer, lung cancer, pancreatic cancer, and we put the biomarkers, then with one drop of blood, you can test for multiple cancers. And if people are doing it sitting at home, no one test is going to be diagnostic, but it can produce anxiety. These will have to be developed, the protocols for what should be done with me if there is a circulating tumor cell, what's the next step I should do, and the next step, and bring insurers and Medicare, Medicaid, etc., to cover those kinds of supplemental screenings that I would need. So these are all, they appear very difficult issues, but let me finish by saying that we would have been looking for a better ribbon for our typewriter, but that has become moot point since the word processor came on. Nobody thinks of a typewriter anymore. So once we have a new thing that comes out and we prove its success, as Grail seems to be doing, as Thrive seems to be doing, Guardant is doing, I mean, the unbelievable technological advances that these young people are bringing out is so phenomenal. I'm very optimistic that instead of provoking anxiety, it will give us the chance to develop more targeted therapies to zap the cancer at its inception.
Isn't the argument that's being made between Guardant and Thrive and you, Dr. Raza, that the bolus of expense that is currently being spent needs to be moved forward? Right?
Yes, yes.
Right? And whereas, you know, what we're doing, and we can look at how the FDA, you know, approves drugs and what's being done on that front, and you know, the amount of money that is being spent to extend someone's life by 3 months or 4 months or whatever, and the toxicities associated with it. I'll throw this out to all the fellow panelists. The argument seems to be that if we can take those same dollars and reallocate them towards earlier, you know, whatever the technology may be, that the reality is that we won't spend more, but in fact, over time, we'll actually spend less with better outcomes.
And Harris, you said it, because I think that's the key piece, right? There's a quality variable that we want to solve for, right? That so much of the emphasis is on cost. And we orphan this quality component of it. And so can you start to do that when you move it up? And exactly as you said it, and I start to value quality of outcomes, quality of treatment, quality of diagnosis more than I do today versus just how much did that cost? And we want to include that. I'm not saying that we're going to obviate the cost question, but we're going to have another variable in there that hasn't been measured nearly as well in this regard. It starts to be in other areas of healthcare, but in the oncology world, quality measure has sometimes gone a little unnoticed, and that would help improve that by shunting the care towards the primary care as well.
Yeah, I think Harris, that you know, you hit the nail on the head though, in terms of we have to really look at net health outcomes benefit, you know, from these technologies, especially when you think about broad screening. And frankly, you know, I think we don't know with some of these technologies in terms of where the sensitivity is and specificity, in terms of what type of cancers they detect. Would that truly, from a long-term point of view, essentially improve net health outcomes? And that's the challenge. I mean, the technology is one challenge that many of us are working on. I would say 5/6 of the challenge, you know, 80% of the challenge is actually getting a technology that works into the healthcare system, changing the standard of care, changing clinician workflows, getting reimbursement, getting into guidelines. All of those things are frankly much harder and a much bigger expense. And the biggest expense is time. You know, I think, you know, what we're doing is we're starting with a single cancer, but then we're, you know, going to multi-cancer, you know, very quickly. It's what we did with liquid biopsy for the metastatic setting, starting with lung cancer, where there is the highest clinical utility, and then expanding horizontally from there to over 100 cancer types. We believe the same thing can happen in early detection, but you really have to pick your beachhead, at least, and we have maybe a fundamentally different kind of point of view. But, you know, we see a pathway in colorectal screening that, you know, I think Exact has laid out that essentially allows us to get into the clinician workflows, allows us to get into guidelines, and most importantly, allows us to get reimbursement, because we're piggybacking on colonoscopy, where multiple studies have shown net health benefit. And so if you can essentially piggyback on a screening technology that has shown long-term clinical utility, then you can go much more quickly. The challenge with cancer types and early detection that don't have any type of screening is you have to really do, you know, I think if you're being honest with yourself, it's a study that is similar to the National Lung Screening Study, There was 100,000 patients, 10 years, $300 million. And, and it was positive, but only marginally so. There was adverse, events of biopsies, a 1% mortality rate because of the biopsies that were done, very high, false positive rate from single imaging. It got better with dual imaging, low dose CT scans, and it was positive, but you have to do those types of studies. We've seen almost every type of screening technology be walked back, whether it's mammography or PSA to active surveillance. And so it's a balance of finding where should we just be watching these patients versus where should we actively intervene. And so we have to really look at that diagnostic and interventional odyssey that's going to be unleashed by these technologies versus the sensitivity that's there and versus the cost to the system. Look, my choir badge is flashing. I'm preaching, for early detection, but the reality is, for any of our companies and for to be successful and for the healthcare space to ultimately be changed, these technologies have to be completely reimbursed, and there aren't, unfortunately, a lot of pathways today for broad cancer testing for reimbursement. And so there's going to have to be some either legislative changes or systemic changes to how reimbursement happens for that to happen within the next 3 or 4 years.
Yeah,
Can I add to that, Ben?
Please do. Go ahead.
What Helmi said, I want to add to it. Listen, these wonderful tests that are starting to come out, we are just at the tip of the iceberg for this early detection approach. It would be very hard to apply them to the population as a whole immediately. One thing we should consider is starts by, I feel like all these various attempts that are being made in a haphazard way. There has to be some sort of integration of efforts for early detection. And I think the two things I wanted to add to help me. Number one is that you were right in saying that 90% of NIH budget, NCI budget goes into trying to chase the last cancer cell down and kill it. And then we end up, as Harris said, by improving survival for a fraction of patients or by a few years at a cost that is financially ruining them. But recently, there was a $1.1 billion increase in NCI funding, and I was so pleased to see, and many of my colleagues started sending me emails immediately saying, listen, somebody has heard you, because almost $690 million of that is going towards early detection measures now. So there is going to be a turnaround, and I can send that to you, Helmi. And the second thing I wanted to say was that yes, this money is going towards early detection. Even NCI is beginning to invest in it, and I think the excitement is picking up. But let's target a population who is at high risk of getting cancer instead of the whole healthy population. Who are they? Cancer survivors. Because you know that 1 in 5 new cancers arises in a cancer survivor. My husband is an example. Harvey got his first cancer at 34 years of age and survived it. At 57, he gets a second cancer and dies. So you just do the numbers. If we have 1.7 million patients being diagnosed with new cancers every year in America, 340,000 will occur in cancer survivors. Every major institution in this country has a cancer survivors clinic. I'm saying we bring the patients in, we do a physical exam, do a couple of CBC and metabolic profile or whatever, and send them home. We should be saving samples on these patients. MD Anderson sees 80,000 cancer survivors a year. At Columbia, we are seeing 1,000 cancer survivors with breast cancer. Why aren't we saving their samples? And if we have their samples, a certain percentage will get cancer. Now we can go in retrospect and examine all these early detection techniques and see which one picked up cancer 5 years in advance or 2 years in advance. This is the kind of approach I'm talking about, and proper integration of all this requires a lot of education. in addition to policy and technology, we need to concentrate on educating everyone about why it's essential to do it. Okay, I'll shut up.
Yes,
I agree with you.
Yeah, thank you. I wanted to just come back quickly to Harris. You raised reimbursement, and Helmi, you mentioned this too. And I'm just curious if the goal is for insurers to cover early detection as part of a standard cancer screening process for healthy adults, or is it more like Dr. Raza suggested, segmenting specific patient populations? And Harris, what do you think is the correct goal, I guess, for achieving reimbursement?
Well, I mean, it would have been great to have had, Michael Ackerman or David Wilcox or someone like that on this panel. But I think what the payers are very sensitive to when it comes to screening is paying twice, right? So in essence, I do a Cologuard, I get a positive, then I have to have a colonoscopy. And so now I've paid twice, right? And or that someone has very early disease and that disease doesn't advance, right? And so I think the issue or the elephant in the room may be, how do we do better risk profiling of patients to be able to say which are the patients that we should be doing early screening on, right? And then make that screening affordable so that, again, because the payers are mandated by Congress that they've got to pay for the drugs, right? They don't have a choice, right? And so, and then you have another issue which hasn't been discussed, which is that, I hate to think this way, but physicians, gastroenterologists make money off of colonoscopies, right? And so, when we talk about reimbursement, it's not just reimbursement for the test, but it's also how do we rewire the delivery, the diagnosis and delivery system, so that we're aligned on the goal of early detection, and get that aligned, across the board. And I think payers are very sensitive to this issue of getting. They don't want to do early screening because they don't want to pay for false positives, and then they still have that expense down the road for the 200,000 cases that do advance.
Well, I think other challenge too, Harris, is payers would rather play musical chairs and hope they're not the ones who get left without a seat. The average individual only stays 18 months with a specific health plan, and so there's not a lot of incentive to try to unearth every possible disease in that individual. Just pass the buck to the next guy is kind of where the industry has been historically.
But there's, Helmi, there's actually really good news on that front, okay? Which is that because of the consolidation of payers, right? You now got 3 or 4 payers that are accounting for 70% of all the lives. So there's less of that kicking the can.
We haven't seen that help because, unfortunately, those 3 or 4 payers are the ones that have hired the most conservative medical directors. And much on the diagnostic side, they'd rather just push the buck and not know rather than pay up front. I mean, even for something like testing of cancer patients for biomarkers that have proven clinical utility, United, Aetna, and Anthem still don't pay for comprehensive genomic profiling. So, unfortunately, we haven't really seen the needle move, very far there.
Sure.
I apologize. I think we could keep this conversation going for quite a while, but we need to wrap up. We're coming to the end here. I want to thank all of you again for a really stimulating discussion. There were a number of questions about, that came in from the audience on specific ideas about education and, policy changes that are needed. So I may just ask all of you offline, if you have any specific thoughts that I can share with the audience via email on that. But we'll end it here for now. And Afamia, I'll turn it back over to you for the wrap up.
Thank you, Ben, and our entire esteemed panel for a great conversation today. As a reminder, a recording of today's session will be emailed to all registrants and available to view on demand. On behalf of Informa Pharma Intelligence, thank you again for joining us and hope you enjoy the rest of your day.
Thank you.
Thanks, everyone.
Thank you.
Thank you. Thank you, and thank you, Informa.